Hello everyone, welcome to my channel. So today we are going to learn about the topic photosynthesis in higher plants. It's a very important topic. So we can see that plants can make their own food, right? And how they are making, they require four things. That is our carbon dioxide, which is entering through the stomata, which is present in the leaves. And then we can see that the water is being absorbed by the root and it is going to the different parts of the leaves, and the leaves are having this green pigment which we call as the chlorophyll, which is absorbing the sunlight. That means these four things are very important, and you can see the light energy is being converted into chemical energy here. So that's why we can also say it is a physico-chemical process, right? And here we can see it's a redox reaction. Why so? Because using the solar energy, we can see that it is going to oxidize the water and forming the oxygen. So we can see there is a oxidation, and we will see there is going to be a reduction as well. So we can notice that carbon dioxide which is present here, it is going to be reduced, okay, and form the glucose. So there is reduction and oxidation. Now you must be wondering, ma'am, why this photosynthesis is an important process? We know photosynthesis is important for two reasons. Firstly, of course, it is making the food for the plant, but it is also the primary source of food for our earth, right? Because we know about the food chain, that plants are the producers, herbivores are eating on them, and then that's how it goes to the upper trophic level, and we are getting also the oxygen. So you can notice oxygen is also released by this photosynthesis process. So one fine day we didn't knew about this process, right? That whole how this photosynthesis take place. So there might be various scientists who has done various experiments. Then they got to know. So in today's class we are going to first know about them. So the first scientist I'm going to discuss about is Joseph Priestley, who discovered oxygen in 1774, and he also discovered that the air which is evolved by the plants only. So it's a kind of air which is going to purify the air which we are breathing. That means he told that when he did an experiment using a mice, he used a bell jar, okay, two bell jar and candle. So what he did first in the set one, he like kept the rat, and then he also kept a candle burning. After some time, he saw that the mice also died and the candle also got extinguished. Then he came up with a like idea, why not introduce a mint plant? So then he kept the mint plant, and he could see that in the set 2, when he kept the mint plant, then he could see that the mice was alive and also the candle was also burning. Then he came to a conclusion. It is that the plants are releasing something, okay? That means what are plants releasing? A kind of gas, that is we know it's oxygen, and when it is releasing, what is happening? It is going to purify the air which have been like made foul by the mice. Like when we are breathing, we are releasing something that is making the air foul. So he has told that when the animals are breathing and burning of candle is happening, it is going to remove something from the air which is going to make the air foul, and the air is going to be purified by our plants only, okay? So plant is going to restore to the air whatever when we are breathing, we are making it into a foul air, we can say. So the plant used is mint plant, okay? Now Jan Ingenhouse, he discovered that yes, air is released, that is the oxygen is evolved, but he has done this experiment using a hydrilla plant, and he also did it once during the sunlight and during the daytime. That means in the dark and the sunlight. So he showed that sunlight is essential, because when he did the photosynthesis, that means he just kept the hydrilla plant, it's an aquatic plant, so it kept it in the sunlight. He didn't like in the dark, he didn't see any bubbles coming. But when he kept it in the sunlight, he could see that the bubbles of oxygen were visible. So he came up with an hypothesis that it showed that sunlight is essential for photosynthesis and plant can only form oxygen in presence of sunlight. Now, further we could see that the like the food that is made by the plant is called glucose. Now, Julius von Sack, he told that the glucose which is made by the plant, it is not stored in the form of glucose, rather it is stored in the form of starch. And back then in 1854, we didn't name this green pigment or the chloroplast organelle as chloroplast. Okay, back then he named that the green substance present in the plants is located in special bonds. He just said this. Later, we have named them that the green pigment is the chlorophyll and the special body is called as the chloroplast. After that, Cornelius Van Niel, he has done a remarkable experiment which also changed our equation of the photosynthesis. You must be wondering how. So he proposed that the oxygen that is evolved by the green plant, it is coming from the water. So we have seen that the that is our water is being oxidized and forming the oxygen. So he also proposed this, and he also told that the hydrogen which is participating in the reduction, so hydrogen from a suitable oxidizable compound is going to reduce the carbon dioxide into carbohydrate. Okay, so if all the the oxygen that is coming, in case of the oxygen that is released, that is coming from the water only. So if you come to the old equation here, so we could see there is only 6 molecule of oxygen, right, in the reactants. But if you come here, when we are balancing the equation, there is 12 atom, right? We can tell there is 12 atom. But in case of the if we come here, let me write it here, it won't be visible. So here we can see only 6 atom of oxygen, and we can see here that there is going to be 12 atom of oxygen. But this can't be, right? Because we know that the oxygen that is coming, Van Niel has proposed, it is coming only from the water only, the oxygen molecule. It is not coming from the carbon dioxide. So the new reaction that we should write, it is the carbon dioxide 6 carbon, 12 H2 is utilized, and it is going to form, you can see now it is 12 atom of oxygen, right? And you can see that the 6 atom is also released. So just to balance the equation, and also to determine that yes, the oxygen that is evolved, it is evolved from the is our water only, we had to do this. Okay, so I hope up till this is clear. So now you must be wondering that we require a hydrogen donor, right, for the formation of the carbohydrate from the carbon dioxide. But this hydrogen donor can be water, and this hydrogen donor can be H2S as well. When it is H2S, that is regarding the green and purple sulfur bacteria, where we can see it is not an oxygenic photosynthesis, rather it is a anoxygenic photosynthesis. So in anoxygenic photosynthesis, the oxidation product is sulfur or the sulfate, but as you can see H2 is utilized, the oxidation product in the green plant, it is the oxygen only. And pigments, you all know that chlorophyll is present and other accessory pigments are also present, we will be discussing. But in case of the green and purple sulfur bacteria, we can see bacteriochlorophyll, that is bacteriopurpurin and bacterioviridin is present. Now, Van Niel only just proposed that the oxygen that is evolved from the green plant, it is going to come from the water, not from the carbon dioxide, right? We only know it is coming from the water. So we have to prove this. For this, we have utilized radioisotope tracer technique. That was done by Rubin and Kaman's photosynthesis experiment, where he used a radioisotope of the oxygen. So radioactive oxygen, it had O18 as the mass number, because 10 neutron and 8 proton, but the usual ones, the normal ones, have 8 number of protons, 8 neutron only. So when the experiment was done, we have seen that the carbon dioxide's oxygen was the normal oxygen O16, but the radioactive oxygen was present in case of the water. Now, after the photosynthesis process, we will notice that only the oxygen which is evolved, it is having the radioactive oxygen. So if the oxygen is only having the radioactive oxygen, this determines that it is coming from the water only. It has not come from the carbon dioxide. So that's how they have proved, and you can see this O16 is going to be present in the carbohydrate. So this proves that carbon dioxide is going to give the oxygen not during the evolution of the oxygen, rather it is giving in the carbohydrate formation. Okay. Now that we are clear, after this, we also understood like necessity of the carbon dioxide, for which the moles half-leaf experiment was useful. So he has taken a destarch plant. After taking that, he has kept half portion of the leaf inside KOH, which is an like carbon dioxide absorbent. So we will see this half part of the leaf will be devoid of carbon dioxide, okay? But the rest part is going to have carbon dioxide. So let's say if we are going to do this experiment, okay? So after this is happening, we have kept it in the sunlight. So if we have kept it in the sunlight, and now let's say we are going to do the starch test, okay? So this is the half portion of the leaf. So this half is inside the KOH, okay? So this is under KOH, okay? This front portion. But outside portion, it is having carbon dioxide. So CO2 is present here. So we can write here CO2 is present. It is not being absorbed. Now, if the CO2 is present, what we have seen that the CO2 is reduced and it is going to form the glucose. That means if CO2 is present, that glucose is produced. So after the glucose will be produced, we will see it is going to be forming into starch, because we know glucose is stored in the form of starch. So we know in the starch test, what do we use? We use iodine. And when there is starch present in any food item or even in the leaf also, this proves that photosynthesis in that leaf has taken place, right? And if in a food item, if we are adding iodine and we can see there is blue-black color, that means that the food item is containing carbohydrate. So if we are giving it in the leaf, we will see if it is turning blue-black color, that means this portion will obviously turn into blue-black color. We are going to notice, okay, this portion. Why so? Because starch is present, so it will show blue-black color. But on the other hand, if we come to this portion, we will see no CO2, right? Because KOH has absorbed. So this tells us that glucose is absent. Glucose is not produced. Is not produced. And if glucose is not produced, no starch. And if it is showing no starch, we will get negative result for the starch test after we add iodine. So it won't show blue-black color like we can see here. So blue-black color will be in the half portion of the leaf where carbon dioxide is present. Okay, blue-black color. Now, can you explain, it is given, can you explain how this conclusion can be drawn? Yes, we can, because the half portion of the leaf is having CO2. If the CO2 is present, half of leaf has CO2. So, it will show positive result. Okay, for what test? Okay, let me move them a little bit away from here so that this problem doesn't happen. Okay, so we can see. So it is going to show positive result. Okay. So we can see it is going to show positive result for starch test. Okay. I hope this much is clear, that there is necessity of carbon dioxide, because we have only seen here that carbon dioxide is going to be reduced and it is going to form the glucose. Now, there was T.W. Engelmann. He also measured the first action spectrum. And what is this action spectrum? So, action spectrum is for photosynthesis and it is constructed for the measurement of the oxygen evolution, because we have seen that oxygen is evolved, right, during photosynthesis. So, but if we want to check the action spectrum, like in which wavelength of light we can see that photosynthesis is happening, so we have to check in which light there is going to be oxygen evolved. So for this, we have taken aerobic bacteria, because we know aerobic bacteria, they require oxygen, right, for growth and respiration. But what we can see here, that we have taken a cladophora filament. So it's a green algae. So once the white light, so we all know that white light, if it is going to pass through the prism, it is splitting into 7 colors, that is of the VIBGYOR. So as it is splitting, you can see that it is starting from, that is violet, that is around 400 nanometer, and it is going and it is around 700 nanometer, that is our red light. Okay, so it is splitting into 7 colors. Now when the light of such wavelengths are hitting the cladophora, what we can notice that photosynthesis will obviously take place, because cladophora has the photosynthesis pigments. Now when the photosynthesis pigment are present, so oxygen will be evolved, and as the oxygen will be evolved in the position where we see the oxygen is going to be evolved, we will see more amount of bacteria in that area. So we could see in the red region there is maximum of the aerobic bacteria is present. So we can say that maximum photosynthesis has taken place in the red light. And also if you come here, in case of the blue light as well, we can see. So what we have observed here, accumulation of the aerobic bacteria in the blue and red light, as these lights are strongly absorbed by the chlorophyll which is present in the cladophora. Okay, so we will be also seeing how the action spectrum looks like. So now before we jump into that, I would be telling you about the different pigments present in the plants, higher plants mostly, but there are other pigments also which are there in other we can say algae or blue green algae, okay, or bacteria. But the pigments, what are those? These are the substances that have the ability to absorb the light at specific wavelength. So we know that chlorophyll, it is like the not an accessory pigment, it is the main pigment. So there is accessory pigment and there is the main pigment as well. So chlorophyll, it is having a porphyrin head and a phytol tail, and it is the primary photosynthesis pigment. But which one is primary? Chlorophyll A. But on the other hand, chlorophyll B, it is not the primary, rather it is a accessory pigment. So what is the accessory pigment? Let's first understand that. So accessory pigment, they absorb the sunlight and transfer it to chlorophyll A and protect the chlorophyll A from photooxidation. So chlorophyll A being the main, like it's like a king, okay? We are protecting our king. So king is being protected by soldiers, by the minister and their kingdom. There are so many, like we can say soldiers, armies, army generals, all are there. So you can say accessory pigments are like that. So chlorophyll B, which is having a formula, you can notice, that is C55, and we can see H10O6N4Mg. And on the other hand, if you see chlorophyll A is having formula C55H10O5N4Mg. So in this photosynthetic pigment, you can see both are going to have porphyrin head and phytol tail. But the side group of chlorophyll A is going to have CH3. On the other hand, the side group of chlorophyll B is going to have CHO aldehyde group. Okay, so this is one photosynthesis pigment, that is chlorophyll. Then we can see that in other plants there can be also chlorophyll, not higher plants, rather the we can say the bacteria, there can be have also chlorophyll D, E, all these are also present. We won't be discussing in today's class. Now the carotenoids, okay, so you can see carotenoids, there are two like subclass, or we can say they are of two types, like the carotene, which is orange-ish color, okay, reddish orange color. You can see the formula C40H56. It is not having any oxygen, rather the xanthophyll, yellowish color we can see. So it is having C40H56O2. So these both are the accessory pigments only, and they are also protecting the chlorophyll from photooxidation. So we can see that chlorophyll is there, and there is also carotenoids, and we can see one carotenoid is having no oxygen like in carotene, but xanthophyll is having oxygen which is present. So now you must be wondering, ma'am, we have seen that the leaves are of green in color. So how we could get these pigments? So if we are going to do paper chromatography of a leaf, if we make a like extract, leaf extract, then we are going to see that all these different colors of pigments will be visible. Okay. So students, we are now going to discuss about certain graphs, which is going to be about absorption spectrum and action spectrum. So firstly, what is absorption spectrum? We know that the pigments, they are going to absorb the light, right? So we have seen that if white light, it is going to split it into 7 lights when it is passing through the prism. So in the T.W. Engelmann's experiment, right? So if we are going to give the pigments, let's say the chlorophyll A, chlorophyll B, and the carotenoids different kinds of wavelength of light, so we are going to see how they are going to absorb. So if you see the first graph which is present, it is going to be absorption of light by the chlorophyll pigment. So at chlorophyll A, you can see it is going to show a peak and blue light, and it is also going to show a peak in the red light. And you can see chlorophyll B, it is also showing the same thing in blue and red, right? But carotenoids, it is not showing, it is only showing its peak in case of a green light only. Okay, you can all check the the wavelengths are written of different light. So we can see that blue light, it is around this region only, because we can see it is around, we can notice after 400, so it is 450 to 495. But if you notice, in case of the chlorophyll A, there is a small peak. So that means chlorophyll A is showing a peak at little peak at violet, and the chlorophyll A and B is showing a peak at blue, and we can see chlorophyll A and B is also showing a peak at red. But carotenoids, they are not showing at the red or the blue light. Rather, it is showing a peak in the red and the yellow region only. Now, if you come to the second graph, it is the action spectrum graph. So the graph of action spectrum, we have seen it was first done by our T.W. Engelmann. So the first action spectrum graph was measured by him. So what is action spectrum graph? It is the representation of the rate of photosynthesis against the wavelength of light. So we can see there are various wavelength of light, right? So as we see that the light is given, it is going to increase, but the increase we can see it is around after 400. So if it is after 400, or we can see it is 450, after 450 only, so we can say it's the blue light. And again we can see a decrease, but suddenly it is starting to increase as well. So it is going to increase at which wavelength? Let's notice it is like before 700, we can say, or yes, we can say it is like around or after before, or we can say around 700, right? So it is around 700 something. So we can tell that it is the red light. It is where it is showing that there is going to be a peak, right? So we can see that the action spectrum is showing its peak in the blue light and the red light only. So what we can conclude from this, students, like if we superimpose the two graphs, so the black one, you can see it is the action spectrums graph, action spectra of what? Of photosynthesis. And this blue color, it is the absorption spectrum, absorption spectra of which pigment? It is the absorption spectrum of chlorophyll A. Okay. Now, if you all see that both are showing peak at around blue light area only, and we could see it is also showing peak at red light. So this tells, as a conclusion, we can conclude that chlorophyll A is the chief pigment associated with the photosynthesis, and photosynthesis takes place in the blue light and also in the red region. Okay, so I hope you can all notice that I won't say it is fully superimposing, but if we superimpose, we can get this idea. Now here the question is, can you determine the wavelength of color of light, at least the chlorophyll A is showing maximum absorption spectrum. Of course, it is the blue and red line, right? We can tell. But the next question is, does it show another absorption spectrum at other wavelength too? Yes, we can see that at this wavelength, that is around the violet color, if you all check it here, around the violet, we can see chlorophyll A also showed a little peak, right? It is showing a little peak. So as it is showing a little peak, we can say yes. And for the next question, yes, in violet light. Okay. So now we can see that these pigments, they are going to absorb, okay, the light. So let's say the chlorophyll, okay, if it is going to absorb the light, what is going to happen? So you can see the chlorophyll when it absorbs the light. So this H mu that you can see, it is basically the light that is telling about, that is the photon. So when it absorbs a photon, and it is going to make a transition into a higher energy or a excited level. That means the chlorophyll is now excited. So this is the excited chlorophyll. So there are total 4 pathways of my excited chlorophyll. First is that the excited chlorophyll can re-emit another photon, okay, re-emit the photon that it has absorbed and return to its ground state. So this chlorophyll is in the ground state, and this is the excited chlorophyll. This can happen, and this process is known as the fluorescence. All right, then we can see by returning to ground state by directly converting the exciting energy into heat and emission of photon. So we don't see this in case of the photosynthesis. We neither see fluorescence, neither we see any heat is evolved or emission of photon. Rather, we can see the excited chlorophyll, it is going to transfer the energy to another molecule. So this is also done during photosynthesis and also photochemical reaction as well. So where is this chlorophyll, where this photochemical reaction takes place, and how does this energy transfer takes place? So everything takes place in an organelle, that is the special body we know. It is our chloroplast. So chloroplast, it is a membranous system. So we can see here, it is a two membrane organelle which is present in the plant cell exclusively. So it is having outer membrane, inner membrane. And just like the mitochondria, it is having a matrix. So here we can see the matrix. It is not called as that. It is called as the stroma. So stroma is the fluid compartment surrounding that is surrounding the thylakoids. So what are these thylakoids? So you can see this disc-like shape. All these are present. So disc-like shape, you can see idli-like of structure. So these are basically called as thylakoid. Now, when they are together in stacks, so stacks of thylakoids are called as the granum. Okay, so granum are stacks of thylakoid, and we can see these granums are collected. Granums are connected to each other. But how they are connected? They are connected by stroma lamellae, or we can also say that is stroma thylakoid. So there are two kinds of thylakoid, that is grana thylakoid or grana thylakoid, and we can see stroma thylakoid or stroma lamellae. Okay. So we can see that inside a thylakoid, we will see there is going to be a lumen. Okay, thylakoid lumen is there, and we have the thylakoid membrane as well. So unlike the mitochondria, we will see all this like different kinds of photosystem or even the enzyme. All these are going to be present in the thylakoid membrane. So the mechanism of the photosynthesis that takes place, it is going to take place in the thylakoid membrane in the lumen portion, and you will see that stroma is also participating, but the outer membrane or the inner membrane, they are not participating. Okay, so you can see I have written here light or photochemical reaction. So if you remember, I was telling that photochemical reaction takes place during photosynthesis. So the light reaction or the photochemical reaction takes place in the grana or the thylakoid or the stroma thylakoids. Okay, grana thylakoid and stroma thylakoid. But there is also something called as dark reaction or carbon reaction. We will be knowing in the upcoming classes what are this, which are taking place in the stroma, that is in the fluid filled compartment that is surrounding the thylakoid. Okay. Now, as I was telling that these pigments are present. So pigments are like watercolors, right? So we don't like keep watercolors like throughout our room, right? Like in liquid stage, we are going to keep it into a container, right? So similarly, these pigments are also kept in a like container. So this container I would call as the photosystem. So what are this photosystem? These are the functional and structural elements of the photosynthetic complexes, protein complexes. They work together out for forming any basic photochemistry of the photosynthesis. So we could see that there was photochemical reaction like light absorption, energy, as well as electron transport. All these are going to take place. Now, in the photosystem, like I was telling you, there is a container. So the container is going to have various compartments. Okay, so there are two compartments we can say. There is antenna complex and reaction center. Okay, so in this compartment, in the antenna complex, it is basically a light harvesting complex which is made up of proteins. Not only proteins, it is made up of chlorophyll B, xanthophyll, and carotenoids. So this is the antenna complex that you can see, and we have the reaction center. So reaction center is like the king. And we have to also discuss about this accessory pigments, which are the part of the axis, and that is our antenna complex. So they are going to collect the light. You can see the light is falling here, and then the light will be transferred to the reaction center. So we have seen that something is brought to the palace. First, it is going to the armies and let's say or the guards present in the palace, then it is going to the ministers are going to give it to the finally to the king. So it is like that. Okay, first the accessory pigments that is present in the antenna complex, they are going to absorb, and then finally they are going to transfer it to the reaction center. But who is the king in the reaction center? It is our main photosynthetic pigment, that is chlorophyll. So after the accessory pigments, they absorb the energy of the photon. We will see they are going to start to vibrate. They start vibrating. So they are going to quickly go to the king to give the anything that they have got, right? We have seen this is happening. So the vibration is passed from one accessory pigment, let's say from one to this one, this one to this one, this one to this one, and finally it is going to come to the reaction center like this, from one to another pigment to another, until the energy is in the vibration is passed to the reaction center. So who is there? Our chlorophyll A. So in the reaction center we have chlorophyll A, which is forming the reaction center. Now, don't think that there is only one kind of photosystem. There are total two types of photosystem chlorophyll, that is photosynthesis happening. So there is photosystem 1 and photosystem 2, which we call as PS1 and PS2. So what do we see? That these pigments are of having two discrete light harvesting complexes, that is PS1, which is having maximum absorption spectrum of 700 nanometer. That's why it is also called as P700. And PS2, it is also called as P680 because it is having like maximum absorption spectrum of 680 nanometer. Okay. So now what is the difference between the two photosystems? So the reaction center, it is going to have P700, and PS2 it is going to have P680. Absorption spectrum is going to be 700 nanometer in PS1, and it is basically the far red light we also tell us, and we have 680 nanometer which is having which is the red light only. This is the absorption peak of the chlorophyll, that is the reaction center. So where it is located? Okay, PS1 we can see it is located in the outer surface, or we can say non-appressed region of the thylakoid membrane. So you can see these are the non-appressed region, that is outwards. But when it is coming inward, so you can all notice in this inward region, okay, these are the inwards region. So here we will see it is the appressed region. So in the appressed region, we will not find PS1, and that is why we are only getting the PS1 in the like we can say only in the appressed region, that is present in the outward region. So it is present in grana and stroma thylakoid. Okay. So it is participating in cyclic and non-cyclic like photophosphorylation. So what is photophosphorylation? We will be studying in the next class. And we can see it is helping in the NADPH synthesis as well. How it is helping, we will be also knowing in the next class. So photosynthesis, photosystem 2, we can see it is present in the inner surface or in the appressed region of the grana thylakoid. So as I was telling, these are the appressed region. Okay. These are not the outer surface, rather they are the inner surface. So the appressed region of the thylakoid. But is the appressed region present in our like stroma thylakoid? This is our stroma thylakoid, right? These are both stroma thylakoid. Do they have any like inner surface or appressed region? No. So that means it is only going to be present in our grana thylakoids. Okay. Only in the grana thylakoids inner surface we can see they are going to be present, and they are going to participate in the non-cyclic photophosphorylation, and it is also helping in the ATP synthesis and hydrolysis of water. So how this mechanism like takes place, like non-cyclic photophosphorylation and there is cyclic photophosphorylation, we will be knowing in the next class. Okay. So have a nice day, everyone. Thank you for attending today's class. Today we have understood about how the photosynthesis takes place, how different scientists have taken like understood about the process of photosynthesis, which are the components or the raw materials important. We have understood about the like in which wavelength of light we will see maximum photosynthesis take place, and we have also understood about the pigments. Okay, so make sure you are revising. Have a nice day, everyone. Take care. Bye.